Welcome to the DRPLA Research Update. My name is Silvia Prades and I am the DRPLA Research Manager for Ataxia UK and CureDRPLA. Today I will be talking about the work Ataxia UK and CureDRPLA are doing to advance DRPLA research. CureDRPLA was incorporated in November 2019 after Paul and Andrea's son was diagnosed with DRPLA. CureDRPLA signed a partnership agreement with Ataxia UK in 2020 to help further its efforts. In our team, we have Paul and Andrea Compton, the co-founders of CureDRPLA, Jeff Carroll, who's a scientist working on Huntington's disease and is our scientific advisor, Julie Greenfield is the head of research at Ataxia UK, I am the DRPLA Research Manager, and Junko Shiozawa is an advisory board member that is helping us expand our work in Japan. Our mission is to connect families, physicians, and scientists to further DRPLA research and work towards the treatment. So let's see what it really takes to develop a drug that can be used to cure, treat, or prevent a particular condition. It all starts with an idea or a discovery in the laboratory. At this stage, thousands of compounds might be candidates. However, only a small number will look promising for further study. Once researchers identify a few promising compounds, they conduct experiments to gather information about its potential benefits, mechanisms of action, undesired effects, and so on. A few drugs will make it to the next stage, which are the preclinical studies. Before testing a drug on people, researchers must find out whether it has any toxicity and which are the benefits. There are two types of preclinical studies, cell studies and animal studies, and we will see this later. At this stage, it can also happen that researchers go back a few steps and redesign some drugs to improve them. Once a candidate has been found, the researchers have to fill in several documents describing their findings and proposing a clinical trial in humans. The paperwork is reviewed by the relevant regulatory agency, which is the entity responsible for approving drugs for human use, and it's also reviewed by the ethics committee. If researchers get the green light, they will start a clinical trial. Clinical trials are voluntary research studies conducted on people and designed to answer specific questions about the safety and effectiveness of a drug. If the researchers have evidence from the trial that the drug is safe and effective for its intended use, they will inform the regulatory agency. And this agency will examine all data on the drug and make a decision to approve it or not to approve it. If the drug is approved, it will be released to the market, and for a few years after the approval, they will continue monitoring the drug for safety and efficacy. Being familiar with the drug development process is very important for rare diseases because there are some additional challenges associated with these conditions. The most obvious one is that they are rare. They only affect a small number of individuals and they can be in different geographical areas. However, efforts have been made to improve drug development for rare diseases in the last few years. The regulatory agencies have produced some guidelines to inform researchers and advocates of what elements are important when developing a program for rare diseases. For today's talk, I will focus on just some of these recommendations, even though we are working on all of them. So let's apply what we just learned about drug development to the DRPLA. But before I move on, I would like to give a brief overview of the DRPLA. You probably know that the DRPLA is a rare form of ataxia that has a higher incidence in the Japanese population. Adult onset DRPLA presents after the 20 years of age, and the most common symptoms are ataxia, involuntary movements throughout the body, dementia, and behavioral changes. On the other hand, juvenile onset DRPLA presents before the 20 years of age, and the most common symptoms are ataxia, myoclonus, seizures, and progressive intellectual deterioration. No one really knows how many people have DRPLA, but we are trying to find as many as possible. DRPLA is inherited in a dominant autosomal manner. This means that there is a 50% chance of passing it to the next generation. It is caused by mutations in the atrophin-1 or ATN1 gene. The ATN1 gene has multiple copies of these CAG blocks. In normal circumstances, the gene produces a normal protein and there's no pathology. However, if these blocks expand and there are 48 or more of these CAG repeats, they will form an abnormal protein, and these abnormal proteins accumulate inside the cells, causing damage, especially in brain cells, and ultimately they will result in the DRPLA. This concept of abnormal protein accumulation in cells will come up later in my talk. So we finally arrived to the most important part of my talk, which is what we are actually doing to advance research. We have developed a research program based on the recommendations by the experts on the field. The first key element that we must work on is to provide a description and understanding of the natural history in DRPLA. The main objectives are to describe the DRPLA population, including a description of all the symptoms and the trajectory of decline over time. In clinical trials, scientists use clinical assessments to measure change in individuals with DRPLA. We need to identify which assessments are best suited to measure change in this community. We also must understand the natural history study is crucial to design clinical trials. We cannot measure if a drug is effective at stopping the progression if we don't understand the trajectory of the DRPLA over time. To address these points, we are funding a natural history study and we created a patient registry. So let's start with the natural history study first. This study has a duration of 3 years and the participants will be visited once a year at one of the sites. There are 3 groups of participants. The adult participants must be 16 years old or over to participate. The pediatric must be under 16, and the remote group is for those that cannot or do not wish to travel to one of the study sites, irrespective of their age. The study doctors will enroll participants with and without DRPLA in all the groups. So, in other words, we have designed the study in such a way that almost anyone with DRPLA can participate. The study doctors will collect the following information: demographics and clinical history. They will collect information about epilepsy if that's present. They will do a physical examination and functional tests. They will evaluate the speech and swallowing function. They will do cognitive assessments, brain scanning, and biofluid collection. It is very important to emphasize that the participants don't have to do everything, and it must be discussed with the study doctors on a case-by-case basis. We have several sites recruiting, one in the UK, particularly in London. We have 2 in the US, one in North Carolina and the other one in New York. But also keep in mind that if you live in another country and you're interested in participating, we can explore that possibility, and these are the contact details for each site. The CureDRPLA Global Patient Registry is a completely different project. This registry is an online questionnaire for people with DRPLA, and you don't have to consult your doctor to participate. This registry is helping the community by characterizing DRPLA, identifying individuals with DRPLA around the world, providing data to scientists reported by sufferers, and also about the burden of DRPLA, and also it will help connect participants with opportunities to take part in research. The study is approved by an ethics committee and it's available in 6 languages. This registry is basically just a collection of questionnaires. For example, it has questions about demographics, diagnosis, medical history, attitude towards research, and so on. We are asking all participants to complete it once a year so that we can track progression over time. As of last week, there are 33 participants in the registry. The youngest participant is 6 years old and the oldest is 67. The average number of CAG repeats is 63, the lowest reported is 50, and the highest reported is 81. Almost half of our participants are of Asian ancestry, and in this graph you can see the number of participants per country. It is very important to get as many people to register as possible because this registry is a great way of showing how many people with DRPLA are there. I know of people in the UK, France, Brazil, Germany, Armenia, and many more countries that haven't participated yet, and we want this registry to be as accurate as possible. With the data from the registry, I can group participants by their age of symptom onset and characterize these two groups. On average, if we look first at the juvenile onset participants, on average, this group experienced the first symptom at 7 years old. And the average number of CAG repeats for this group is 66. There were 3 signs that were equally chosen by participants as the first one they could attribute to the DRPLA. These are delayed developmental milestones, epileptic seizures, and balance problems. When they were asked about symptom progression and current health concerns, the ones that were chosen the most were balance problems, coordination problems, and epilepsy. 79% of our juvenile participants have epilepsy. So now let's look at the adult onset group. On average, they experience the first symptom at 43 years old, and the average number of CAG repeats for this group is 59, so slightly less than juvenile, which is similar to what has been reported in the literature. Balance problems and trouble walking were the signs attributed to the DRPLA onset in the adult group. When they were asked about symptom progression and current health concerns, the ones that were chosen the most were balance problems, coordination problems, mood swings or anxiety, and personality changes. Only 20% of the adult participants have epileptic seizures. This table is just an example of what I can do with the data from the patient registry. We will never divulge any identifiable information, and we will always share the data in a way that prevents identification of individual participants. So let's move to the next topic. Researchers must understand what flows trigger the DRPLA at the cellular and molecular levels, which is just a fancy way of saying that they must understand what has gone wrong in order to develop drugs that target these defective mechanisms. Researchers study these defects in cells and animal models because they can easily be manipulated in the lab. So let's see what we have done and what we are doing to address these objectives. A group of researchers collected skin cells from 16 individuals with the DRPLA. These skin cells have been transformed in a number of ways to make them look and behave like brain cells. It is very important to have cells that are similar to the human brain cells in the laboratory because protein aggregates that are present in the DRPLA are very toxic to brain cells and they can cause a lot of damage to the brain. And researchers must find a way to study this in the laboratory. These cells were developed a while ago and they have been shared with a number of researchers. Studying the DRPLA-derived cells allows researchers to study the defects that constitute the DRPLA and determine if these are reversible. These cells can also be used to test drugs and check if they could be beneficial for the DRPLA. The images shown here are just an example of how these cells look in the laboratory. On the left-hand side, we have an image of cells from a person without DRPLA. Each green circle is one cell. And then on the right-hand side, we can see a similar images, and these very small red circles are aggregates of the mutated ATN1 protein. And as you can see, these very small red circles are not present in cells that do not have DRPLA. Here is an example of how cells can be used. Each line corresponds to the activity pattern of one cell over time. On the left-hand side, we can see that there are waves of activity in these cells, and these seem pretty random. However, on the right-hand side, we can observe that in DRPLA cells, these have synchronized, and they all have bursts of activities at the same time. So let's talk about the mouse models that we have developed. Mice are biologically similar to humans, and they have been used in research for many years. Using genetic engineering, researchers have inserted the human ATN1 gene into mice, and they added 112 CAG repeats so that the mice would develop DRPLA. These mice are being used to test drugs and also to follow the progression of DRPLA from very early birth to an advanced stage. These mice with 112 CAG repeats mimic the human form of DRPLA. They have the mouse version of ataxia, especially in the back limbs. They present seizures and tremors, and they have poor coordination. We don't know if these mice also have cognitive symptoms, but we are gonna, researchers are gonna study this in future projects. This is just a great example of how affected these mice are. So scientists place a mouse at one end of a beam, and they time how long it takes for each mouse to walk to the other end of the beam. On average, a control mice with no DRPLA only takes 8 seconds to walk along the beam. However, the DRPLA mice take 61 seconds to walk from one end to the other. CureDRPLA is funding a lot of research projects that are identifying drugs and studying the drugs' mechanisms of action. This research hopes to develop a drug for DRPLA in the fastest way possible. However, researchers must obtain basic information about the safety and the efficacy of a drug in the laboratory before testing it in humans. They also want to understand how reversible some of these defects are. To address these questions, we are currently working on 2 different approaches: the ATN1 lowering strategies, and we are also exploring drugs developed for other conditions. The ATN1 lowering strategy consists of developing drugs that can block the ability of a certain gene to make a protein. Earlier in the talk, I explained how the CAG repeat expansions trigger abnormal protein accumulation in cells. So with this lowering strategy, what researchers are trying to do is to develop some molecules that will bind to the ATN1 gene, and they will prevent protein formation. And therefore, there will be no aggregates in these cells that can cause toxicity. In DRPLA, we are interested in preventing this abnormal protein accumulation to reverse or slow down DRPLA progression. Antisense oligonucleotides, or ASOs, are a type of silencing molecules that can prevent protein formation. A group of researchers at Boston Children's Hospital have generated ASOs that are capable of reducing human ATN1 levels. Just so you can have an idea of the scale of the project, they designed approximately 50,000 molecules. Only the top candidates are being further validated in cell and animal models. On this graph, we can see some results of the ASO screening in mice. On the y-axis, we have the ATN1 levels in brain cells of mice, and these are around 100 units in control mice. However, when these mice were treated with ASO1 or ASO2, the levels of ATN1 decrease in the brain cells of these mice, which is our main goal. ASOs are our primary focus of attention because they have been approved for human use in other neurological conditions. Therefore, there is precedent that they work in humans. Another type of silencing molecules are called siRNA. These compounds can also successfully decrease ATN1 levels in cell models and brain cells from mice. This is shown here in this graph. In control mice, so the red and the green box, the levels of ATN1 in brain cells are around 1 unit. However, in mice treated with the siRNA, the levels of ATN1 were reduced by half. Although these results are very exciting, siRNAs have one disadvantage. They have not been approved for human use in other neurological conditions. Therefore, we are not pursuing this strategy because it hasn't been widely tested. However, this is what scientists call a proof of concept experiment. It's good to know that what we are looking for is achievable. The other avenue that we are exploring is using drugs that have been approved or are being considered for other conditions. Omaveloxolone is a drug that is being considered for human use by the Food and Drug Administration in the US for Friedreich's ataxia. CureDRPLA is funding a pilot project to test if OMAP could be beneficial for DRPLA in cell models. The project only started in July, and we will report the results in a few months. A big part of our research program involves the participation of individuals with DRPLA, caregivers, and advocates. We want to gather information from people with DRPLA and caregivers because they are very knowledgeable about the condition. Understanding what is important for the community will also guide us during the drug development process, and it will ensure that we meet the needs of this community. We are approaching this in different ways. We conducted interviews with individuals with DRPLA and caregivers. The patient registry also fits nicely with this area of work, and we are also doing a lot of work on social media to engage with the community. Casimir conducted some interviews for us to inform the selection of assessments to use in clinical trials, and also to explore patient and caregiver goals for treatments and clinical trial participation preferences. They interviewed a number of caregivers and people with DRPLA. The main findings from these interviews will be published in a scientific journal soon. I will only highlight a couple of findings from these interviews. The two people that were interviewed with adult-onset DRPLA would like to see an improvement in ataxia, and they would like to stabilize their condition so it doesn't progress anymore. The caregivers that were interviewed reported that they would like to see an improvement in self-care, mobility, and personality to remain intact. We use social media to disseminate our work and engage with the community. I want to highlight the DRPLA Facebook group with 130 members because it has been very active lately. Some interesting facts about us. The CureDRPLA website is most active in Japan and South Korea. Ataxia UK and CureDRPLA have been featured twice in the newsletter from the Japanese Association of Ataxia Patients. Junko, our advisory board member, is very active on social media. She has a YouTube channel and a blog with thousands of views per day. For us, social media is a way to communicate with the community and to provide updates of our work. If you have any suggestions of how we can improve our services or provide more information, just let us know. We want to make sure that we are using these tools in the best way possible. I cannot finish this talk without mentioning the first ever DRPLA conference that was hosted by CureDRPLA in May. This conference was a huge success, which is a great evidence of our momentum. 24 people attended in person and 7 more joined virtually. It was a fantastic opportunity for community building and to share knowledge across different projects. We are so pleased with the results that we are planning to make it a recurrent one. I hope that after today you will have a better understanding of our work. We have achieved a lot since 2020, and I thought it would be nice to measure the impact we are making. From November 2019 until March 2020, CureDRPLA had funded $3.3 million in research. By the end of the year, we will have awarded at least 13 research grants, and more than 50 scientists have participated in DRPLA research. CureDRPLA and Ataxia UK are also working with biotech companies that are doing some research for us. And our impact is global. We have research that is currently taking place in multiple countries. I wish you all leave this talk with one message. CureDRPLA and Ataxia UK are facilitating DRPLA research by enabling safe and effective drugs toward clinical trials and increasing their likelihood of success, all while we are also trying to move forward as fast as possible. And with this, I get to the end of my talk. Here are my contact details. Feel free to reach out to me if you have any questions. And thank you very much for your attention.